Hi, everybody. It's Dr. Campbell, and I'm back with you to talk about what we're doing in biology this week. And this week we're focusing on the Elodea Photosynthesis Lab. Let me explain how this lab is a little different than what the labs we have done so far in photosynthesis. So far, we have looked at autumn leaf chromatography, and we've looked at chlorophyll and xanthophyll and the different types of pigments that are in leaves. So we extracted those pigments, and we know that in the fall, when the chlorophyll is starting not to be used anymore because the leaves are starting to change color, then the photosynthesis process is stopping at that point in time. What we haven't really looked at is rate of photosynthesis. So what we're going to be studying in this lab with the Elodea, which is the same plant that we have used in the past in lab, for leaf, looking at the chlorophyll in the leaves and the chloroplasts, we're going to use this now to look at rate of photosynthesis. So let me explain how we're going to do this. When I set up the lab for you, let me move my picture over here a little bit. I have the Elodea is placed in a glass funnel inside this beaker. So this is a 1,000 milliliter beaker of water. The Elodea I took out of our pond tank, so it's our same Elodea that we've always used. I didn't add anything to it. It's just the Elodea in water. And then over here you can see a 10 milliliter graduated cylinder. And the reason why I'm using a 10 milliliter graduated cylinder is I want to be able to measure my rate of photosynthesis. And in order to do that, I have to be able to collect something. And if you think about photosynthesis and you look at the equation for photosynthesis, a product that comes out of the photosynthesis equation, and some people would say it's a waste product, but the product that comes out is oxygen. So I want to be able to collect oxygen during this lab. And in order to do that, I need to fill this up with water. I'm going to put a plug in it, and I'm going to invert it over the top of this glass funnel right here. And then when the photosynthesis occurs and oxygen is given off by these leaves, it will go up into this graduated cylinder that is now upside down on here. And basically what happens is I have what's called water displacement. So as the oxygen goes up into the tube, it displaces the water will be pushed out, and the oxygen bubble will form on the bottom of this because it's going to be inverted. All right. So here are all the Elodea ready to go. I have tons of it in here. So I have made 6 of these, and I'll explain why I did 6 of these in just a minute. And then you can see there's a graduated cylinder upside down on top of that funnel. And this is underneath a blue LED light. So what we're going to look at is how colors of light affect the rate of photosynthesis. We know that photosynthesis will occur, but what we're trying to figure out is how would the color of the light affect it? So would red light versus blue light versus green light make photosynthesis go faster, and as a result give me more oxygen? And that's what we're going to be looking at. And the reason why, again, we use LED light and not regular like Christmas lights or something like that, except if they're LED, is because LED lights will not give off heat. And heat could have a negative effect on plants if it's way too much. So we're just using LED light because they're cool lights. All right. Then what I do is, you saw that picture, I wrap the entire system in foil. Now you might say, why would you want to do that? Well, if you go back and you look at this other slide, this LED light is coming in, but so is all the lab light and the light from the window back over here. So I don't want just LED. I want just the LED light to affect it. So I'm going to cover up the whole system with foil. So only the blue LED light is coming down on top of this particular plant onto the Elodea. Okay. So then I set them up all over the room like this. I have one in the window just with regular light. I have yellow here, I have red here, blues here, green here, and back behind on the other lab table, I have a white LED light. And then I also have one that has no light on it at all. So when you think about this, when you think about when we run an experiment, we have what's called an independent variable, a dependent variable, and a control variable. So this is where you're going to want to probably write down some notes in your notebook as we go through this, because you're going to need this information for your lab that you're now getting ready to set up. Okay, so we're going to have two parts on this lab. We're going to have the preliminary lab and the lab discussion, and then we'll have the lab data collection, which is the setup ready to go, right? So when I talk about this lab, I'm going to have the independent variable, what I have changed. And what I have changed in this particular lab is the color of light, because that's what I'm testing. I want to know if the color of light is going to have any effect whatsoever on the rate of photosynthesis. So again, I've used blue, green, yellow, red, white, and then regular light, which is the sunlight one, and then no light. So, but it's just really the color of the light. So my independent variable, what I've changed is the color of light. Dependent variable is what I'm going to measure. And if you remember, if you go back and look, I'm waiting for the oxygen bubbles, which you can see they've already started here. They're going to move up through this funnel, and they're going to collect in the top of that graduated cylinder. And I might get 1 milliliter, I might get 2 milliliters. I don't really know what I'm going to get as far as oxygen. I'm going to let this run until Thursday, and we will see what we get. After a couple of days, we'll see how much oxygen we get. So my dependent variable will be my oxygen that is being given off, because I can volumetrically measure that. Now, could I just count bubbles? Sure. There are simple ways. Lots of people, when they do this lab, they just look for bubbles. But I wanted to do it so we could actually collect real data, so that we could make graphs and things like that and understand our data a little bit more and make it a little stronger as far as an experiment is concerned, okay, or an activity, a lab activity is concerned. All right, so I have all this set up. Dependent again is the color, independent is the color, dependent is the oxygen that's being given off. I'm measuring that volumetrically in milliliters. Now we're going to talk about control. Whenever we have a lab, we talk about, or an experiment, we talk about control. Most people think that means what I kept the same in the experiment. And what they might say is, when they look at that, they might say, well, you kept the water the same, you kept the funnel the same, you kept the graduated cylinder the same. That's correct, but that's not the control. The control is when I compare it to something that we know. So for example, in this one, I have a positive control, and the positive control is the Elodea that's sitting in the same system, but it's sitting in front of the window for sunlight. I know that Elodea will photosynthesize in sunlight. So I put it over there. I don't know how much oxygen I'm going to get, but that will be my basis to compare to. If I get a whole bunch of oxygen sitting in the sunlight, that would make sense. If it has a lot of sunlight over the next few days, then that could be the basis that I compare to. That would be normal. So what would happen if my yellow light gives me more than that? That would tell me that the yellow light would give me more oxygen than perhaps any of the other colors. So that's what I'm going to compare to. The other hand, I'm also going to have, in this case, what's called a negative control. A negative control is a control that I don't think it's going to work. I don't think I'm going to get any oxygen out of it. And you've probably guessed what that would be. It's the one I totally covered up in foil. I'm not exposing to any light. I just left it on the counter, totally covered it in foil. I'm not expecting any oxygen bubbles to occur. I might get some. I might get a few that, because the plant was already going through probably photosynthesis, what was sitting on the table, waiting for me to put everything together. There's a possibility that I could have some oxygen come out of that process. Even though it's sitting in the dark. So we'll just have to see. We'll have to see in a couple of days if I get any oxygen from that system. But in general, that will be the negative control. So again, my independent variable is my colored light. My dependent variable is the amount of oxygen in milliliters that I'm going to measure. My positive control will be the Elodea that I put in natural light. And my negative control will be the Elodea that I totally covered in foil and is not exposed to any light whatsoever. And that's important for you to know, because when we put together the lab, you're going to have to understand that information. So now what we're going to do is we're going to skip over and we're going to go to the lab so that you can see what we're going to have here. Okay, so X out of here. And now I'm going to go to the lab. The lab that you're going to open up after your video. So once this video concludes, there will be a timer set and this lab will open up. And so this is on Google Doc. And this explains, again, the process of photosynthesis, how the plants take up carbon dioxide and use the energy of the sun to produce sugar, but it will also produce oxygen as a byproduct. It will talk about chlorophyll, and it talks about light-dependent and light-independent reactions, which you've already watched 2 videos about, okay? With that in mind, we have watched 2 videos, the Bozeman Science video, which is here, and the Amoeba Sisters video, which is here. If you feel like you need to go back and re-watch those videos to make sure that you understand the information in them for the rest of this lab, that's why this link is a live link, and it will take you back to those videos. That's fine. I totally understand that. So I said, re-watch these if you need help. So here are the questions that you need to do before we finish this lab. Right here, you're going to write down the photosynthesis reaction. And it says, make sure you label it, each chemical in the Google Docs. You can format superscripts. Be sure you represent the numbers correctly in this format. So what does that mean? Well, up here, I know that I'm going to produce sugar in the photosynthesis equation. These videos and your book has a photosynthesis equation. But I'm going to show you, if I type in C6H12O6, that is the formula for glucose, which is given off in this particular equation, in this reaction. However, it's not formulated correctly. So let's look how we formulate in Google Docs. How do you formulate this particular item? So if you look down here, I'm going to highlight the 6, and I know I have to make it a subscript. So I'm going to go up here under Format, not the text part. I'm going under Format. And when I click on Format, if I click on text, then it says Subscript, and it automatically makes it a little 6 here. And I do the same thing on the 12 that you can see here. Again, I'm going to Format, going to Text, Subscript. And there's my little 12, and I'm going to do the same thing on the 06. Okay, so that's what I mean by formatting. So in the equation, which you need to write down the entire equation, blank plus blank plus blank yields, and you know you're going to have oxygen, and you know you're going to have glucose. It also says make sure you label by name. So if this is in the equation or CO2 is in the equation, you need to write glucose next to it in parentheses. So just like this, glucose, or sugar. Sugar will work too, okay? Now, I'm going to show you something that could mess that up when you're typing it. If I make this a subscript, okay, and I've made it a subscript, and then I'm going to type in the word glucose. Glucose just became a subscript as well. So if that by chance happens, you can leave it like that. I can read it pretty well, or you can highlight it and just make it go back to normal text. So just make it go normal. That's fine. Okay. But you need to label each word. That may mean you may have to go this plus this yields, and then you may have to do a second line to fit it all in the space. I fully understand that. Okay. Now, what else do you need? You'll need a plus sign, which there should be a plus sign on your keyboard. And you'll probably need a yield arrow. They don't usually use equal signs in chemical equations. Usually we talk about a yield arrow. And so how I do that is up by my plus sign, I do a dash, dash, dash. And then down at the bottom above I have a period and a comma on my keyboard. I have a little arrow above my period. And then I can do that as well. I can also probably insert some picture somewhere, but this is the easiest way to do it. And then I don't have to worry about space. Okay. So now that I've done all that, in this video, Amoeba Sisters, they talked about light-dependent reactions and light-independent reactions. After you put out your equation, you're going to go back and you're going to highlight. So let's say I have the word sugar here. Okay, and I'm going to show how to do this. In that same equation, it says go back and use the highlighter option in Google Doc and yellow highlight the terms from the light-dependent reactions. So let's say this part, this is not correct, so please don't copy this. This part is the light dependent. So I'm going to highlight it. I'm going to go up here to the highlighter. I'm going to choose yellow. Then that part now just became highlighted. Then it says in that same equation, go back and use the highlighter option and green highlight the light independent reactions. So again, I'm going to go over here. I'm going to highlight. I'm going to go up here to highlight, and I'm going to choose green. And then I've done what 2 and 3 have told me, all within this equation up here that I've already written. Okay, so now I'm going to erase this so it's out of our way so we don't have to think about it anymore. Then it says down number 4, where do plants get the CO2, that's carbon dioxide, for this process? Hint, what produces CO2? Provide at least 3 examples. There are probably lots of examples in our book that you can go to. And again, we're in 8.2 and 8.3 on photosynthesis. But if nothing else, you can look it up on the internet. I don't have a problem with you using Google. Search it. What gives off CO2 that plants can use? So you can look it up just like that and list those 3 things down here below. All right. And so make sure you choose wisely. Don't choose things that don't make any sense. And if you're not really sure, then don't use it. All right. So that's all going to be part of your pre-lab questions. And again, you can use your books and your resources. You're going to have some other questions down here too. Where does this occur? Be specific. So not just in the leaves. And number 6 has a question. And then this is the lab setup. So this is basically how I told you how I was going to do it. Instead of a test tube, I use a graduated cylinder. Here's my light source, although mine is on top so that I could cover everything with foil. Traditionally, the lab setup also used sodium bicarbonate, which is baking soda. Baking soda can give me a little extra CO2 for the process to occur. But I'm going to choose not to use sodium bicarbonate. I did not put any sodium bicarbonate in this system because I just wanted to see if the LED works without it. So we used red, green, blue, yellow, and white LED lights. Then what you're going to do is you're going to go to these websites here and you're going to determine which of the lights we use provides the highest rate of photosynthesis. So this website and this website is live, and you're going to read through that, or you're going to watch through that, read through it, I guess, and determine, would the red light be the best? Would the blue light be the best? Which is going to give me the highest rate? And then down here, you're going to say I'm going to say white. If white colored light is used, then the Elodea will have a greater rate of photosynthesis as evidenced by oxygen production. This will happen because. So you have to answer this part, you'll have to answer this part, and then you're going to have a because here that you have to answer. And then go below that and you'll have a couple more questions that will talk about the experiment that you have to answer. And then after that is the data table. You cannot do anything with the data table. So let me scroll down to the data table itself. When you get to the data table, you can't do anything about this because we are currently collecting all the information for the data table. So there's nothing you can do about it. So you'll have to wait until Thursday to see how much oxygen we collect to put on here. Your job is to complete everything for this lab up to the data part, and then stop when you see data next time we meet in class. Just stop right there and don't do anything else. But you need to have everything else done on your lab by Wednesday. All right. I'm going to send this Google Doc into Google Classroom, and I'm going to give you a time of when it's due. So that means you're going to, if you have to rewatch the videos, then rewatch the videos, and you'll have to read the information, and you have to return it to me. Even though this part is not done, I want to read your pre-lab work. I want to see if you understood. Now, if you have to rewatch my video, rewatch it so you can write down the information, because some of the things I said are on this document and I just didn't show you yet. So you need to kind of keep that in mind. Okay. So I hope you found this helpful. When this video is now over, this Google Doc will become live. You're going to open up the Google Doc and you'll have my video. It's also on my YouTube channel. And you're going to answer everything up to the data part and then stop and submit it or return it to me so I can check it. Then on Thursday, we will open this back up when we're in lab, and we will look at the data and what's going on with the oxygen and all these little plant things so that we have a better understanding. We're also going to look at how to put it into a bar chart and a Google spreadsheet. And we'll talk about it more on Thursday when we come into school. So what's due? This is going to be due. This particular thing is going to be due up to the data part, and it will be due on Wednesday. So I hope you found that helpful. I will see you in school on Thursday. Take care.